All right, it looks like we've got a good number assembled already this morning. Thanks so much for joining us, everyone. My name is Yates Coley. I'm an associate investigator at Kaiser Permanente Washington Health Research Institute, an affiliate faculty in the Department of Biostatistics at the University of Washington School of Public Health. I'm also a member of the 2023 class of the COPSS Leadership Academy. As a little background, COPSS is the Committee of Presidents of Statistical Societies. COPSS bestows many awards each year, including the COPSS Emerging Leader Award. From this emerges this webinar series, which is co-organized by COPSS and the wonderful partners at the National Institute of Statistical Sciences, or NISS. These webinars are an opportunity to feature leaders throughout the discipline, invite them to tell their leadership stories, and share their insights. It's also an opportunity to celebrate statisticians and data scientists who have achieved great things and who have inspired others in many different ways. So the topic of today is statistical leadership in research with electronic health record, or EHR data. EHR data refers to data generated in the process of healthcare delivery and includes clinical data recorded at the point of care, as well as insurance claims data that document care received and reimbursed. Widespread uptake of EHRs to store patient data present innumerable research opportunities to understand and improve clinical care, as the real-world data reflect an inclusive patient population and typical patterns of care. As EHR data is observational data not collected for research purposes, research with EHR data presents many statistical challenges. So we're fortunate today to have two of our own here with us who've led in this area. I'll introduce them, and then they're going to give some short presentations and overviews on their leadership journey in this area before we have time for a panel discussion. So we'll start with Dr. Rebecca Hubbard. Rebecca Hubbard is a professor in the Department of Biostatistics, Epidemiology, and Informatics at the University of Pennsylvania Perelman School of Medicine. Dr. Hubbard holds a master's in epidemiology from the University of Edinburgh, a master's in applied statistics from Oxford University, and a PhD in biostatistics from the University of Washington. Among numerous honors, she was elected Fellow of the American Statistical Association in 2019, awarded the Mid-Career Award from the ASA's Health Policy Statistics section in 2020, and received the 2023 Gertrude M. Cox Award from the Washington Statistical Society. Dr. Hubbard's research focuses on the development and application of methods to improve analyses using real-world data sources, including electronic health records and claims data. Thank you so much, Rebecca, for joining us, and we're looking forward to hearing from you.
Thanks for the invitation. I'm really happy to be here today and share a little bit about my journey and my thoughts about how statisticians can and almost certainly should lead in the area of research with EHR data. So I just wanted to tell you a little bit about how I got into this area, why I think it's interesting, and what I think we as statisticians can contribute. Yates mentioned a little bit of my background. It's pretty interdisciplinary. So unlike a lot of biostatisticians, I kind of started out in biology, then transitioned into epidemiology, and then into statistics and biostatistics. And so that contributes to my thinking about how we contribute to EHR research, in that I think understanding the context and the science and being willing to engage on a really meaningful level with the content area experts is really important. But to say a bit about how I got into this in the first place, as I was doing my PhD at University of Washington, I was always kind of interested in and fascinated by the idea of what I think of as found data. So data that we kind of found somewhere, they weren't collected for the specific purpose that we want to put them to, but we would like to repurpose them in some way. And at the time when I was doing my PhD, which was a little bit early relative to the recent interest in EHR data, that wasn't so much a thing, but people were thinking a lot about, if we've invested a lot of money in cohort studies, in big longitudinal cohorts, how can we reuse those data to study outcomes that maybe weren't initially envisioned? Or how could we bring those data together with, say, surveys that have been conducted and try to answer new research questions? And I was really interested in that concept of, how can we kind of use data in ways that are orthogonal to the purposes that they may have been collected for? So that's kind of what was going on in my head when I got my first real job after graduation, which was at the Group Health Research Institute, which has now become the Kaiser Permanente Washington Health Research Institute back in 2008. And at that time, interest in EHR data, it definitely had been there. People had been doing research with medical claims data for decades, but the upsurge in interest in EHR data was really starting to take off. And working in an environment where I was like embedded inside of a healthcare system was incredibly informative and helping to develop my thinking about what we should be doing with EHR data and how we could be doing it. So there was this big surge in interest. I remember going to ENAR and there were just like no end of sessions on comparative effectiveness and how we could answer different research questions using EHR data. And I remember listening to these and thinking like, I think that maybe some of the people doing this research don't realize, because they don't have hands-on experience with EHR data, how really, really messy and complicated it is. And so being very hands-on and close with the data got me very interested in data quality issues and trying to figure out how we can deal with the missingness, the measurement error, the complex observational processes that I knew were going on with these data, and which I felt were kind of foundational and needed to be done before we could answer all of these really interesting comparative effectiveness and causal inference questions. So with all of that kind of in my head, I had two experiences doing research at Group Health / Kaiser Washington that were really formative for me. The first was working on a mental health services research project where I came in as a statistician on this project. It seemed very straightforward. We wanted to know whether or not the amount of money that was left on a patient's deductible influenced whether or not they came in for psychotherapy after making an initial call to schedule psychotherapy. So we thought maybe patients who had more left in their deductible and would actually have to pay for this service out of pocket were less likely to actually show up and receive care. And what we discovered in working on this project was that even being part of the health insurance provider, it was almost impossible to tell at a particular instant in historical time whether or not a patient had met their deductible. So I realized that EHR data are not only complicated and messy, but they're dynamic. They're constantly being overwritten and rewritten with new information. Things were going back in time and changing what should really be historical information. So that made me realize we can't treat these just like any other data source where you can kind of freeze them once and the data are static and whatever happened in the past is kind of locked in. We have to be aware that they're being created in response to complex needs of healthcare administration and healthcare delivery, and incorporate that understanding into the way we analyze the data. The second kind of formative experience that I had was working on a project looking at harms and benefits of repeat screening mammography. And in this one, we were following women over time longitudinally and looking at their receipt of mammography and the outcomes of that receipt of mammography. And we wanted to apply kind of a cumulative incidence estimator to these data. But what we realized was the people who had more mammograms, who we observed sort of longer into follow-up time, were systematically different from the people who had been censored earlier because they had fewer mammograms. And so this was a really great opportunity for statistical innovation because we were able to develop new statistical methods that addressed this informative censoring project. And what I learned from this was I worked on it for quite a while, developing a method to deal with the censoring bias that was inherent in this, this context. And I got a really nice biometrics paper out of it. So that was great for me as a statistician. But on the applied side, when we went to answer the scientific question, it was really motivating the research. The censoring bias model that I had developed required too many assumptions. It was too complicated. It was too difficult to translate. And so we ended up actually using a simpler approximation to that method in that applied publication. And subsequent similar studies have also used that simpler approximation. I'm not sure how many times the more complicated method in biometrics actually ended up being cited. So it was kind of a nice opportunity for statistical innovation, and many of those exist in research using EHR data. But it also really taught me that answering the right question and doing it in a practical way with methods that can be translated and disseminated easily is at least equally important. The last couple things I wanted to say about my research in this area are really about the concept of claiming a seat at the table. And I feel like that is the process that I've been trying to engage in for about the last 15 years to a decade since finishing my PhD. I think statisticians have so much to contribute in this area because of the way that we think about study designs, the way we think about bias and valid inference. But what I've observed is that statisticians often shy away from that responsibility because we're nervous about how messy the data are, and we don't want to make all of these assumptions and kind of make definitive statements about how the data should be analyzed or not when we feel like we don't fully understand them. But I think the insights that we can provide are at least as good as anyone else. Others who don't have training in statistics, our colleagues in informatics and computer science, who are often much more willing than we are to be bold. So over the past few years, that's something I've really been working on, is stepping up, claiming my expertise, claiming my seat at the table, and informing these discussions. One way that I did that, I think, was super powerful, was I had the opportunity through the ASA Council of Chapters Traveling Short Course Program to travel around to ASA chapters all over the country and deliver a short course on analysis of EHR data. And at first, when I started doing that, I felt a little bit abashed because I was like, there are so many people who know so much, so many statisticians who are really, really smart and talented. Am I the right person to be doing this? And then I thought, yeah, I think I am, because I've been doing this for a long time and have a lot of practical experience and expertise. And I think that I have the ability to at least improve, at least move forward the state of the art in terms of the approaches that we're using. So I think when we have opportunities like that to disseminate our knowledge and our understanding, we really need to claim them. So the very last thing that I will say, and then I think my short time is up, is just a little bit about what I'm currently working on. So since my early days working in a healthcare system, I left that environment, and I'm now working at University of Pennsylvania in the School of Medicine here. But most of my research still uses EHR and medical claims data from a variety of different sources. I focus on cancer care, but one of the things that I love about working in this domain is that the lessons that I learn in how to analyze EHR data are really widely transferable. So I also work in cardiology, dermatology, infectious disease, ophthalmology, like kind of across the board here in the medical school, which I find really, really fun, and I feel like creates a valuable contribution to a lot of different clinical areas. The other thing that I really like about the environment I'm currently working in is that I have the opportunity to train the next generation of biostatisticians. So for instance, right now I'm teaching a course on design of observational studies, where I really try to stress to students the importance of understanding the scientific question up front and having that inform all of our design and analytic choices, and balancing practicability of the methods that we choose with valid inference. We always want to minimize bias to the greatest possible extent, but almost all choices involve practical trade-offs, and sometimes the truly unbiased method is not actually attainable. And then the other fun thing that I get to do here is work with medical fellows who are pursuing careers in academic medicine and help teach them about how to work with statisticians, sensible and non-sensible things that they can do with EHR data, and really do some impactful research in terms of answering clinical questions that the next generation of sort of top doctors are interested in asking. So I think that's all I have to say. I look forward to answering your questions.
Great. Thank you so much, Rebecca. I kept like smirking, and I hope it wasn't distracting, but it was just like, I was like, oh, I know that problem. I know. Like, yes, are you in my statistical nightmares? Great. Yes, I too know that problem. So yeah, I really appreciate hearing from you. Next, we're going to hear from Dandan Liu. Dandan Liu is an associate professor of biostatistics at Vanderbilt University, director of the Vanderbilt Institute for Clinical and Translational Research Methods Program, and co-director of the Design and Analysis Unit for the Vanderbilt Biostatistics Data Coordinating Center. Dr. Liu received a master's in statistics from the University of Missouri-Columbia and PhD in biostatistics from the University of Michigan. Dr. Liu has been recognized by her department for outstanding mentoring contributions. Her research interests include biomarker evaluation and event history data analysis with applications to cardiovascular disease, cancer, and Alzheimer's disease. She focuses on statistical method development for risk prediction modeling and its practical implementation in electronic health record systems. Welcome, Dandan. Thanks for being with us today.
Yeah, thank you for the introduction and the invitations. I'm truly honored to share my personal experience with you. So I'm going to share, prepare some slides. I'm going to share. So can you see the slides?
Yes.
Okay, good. So firstly, this is just like my career timeline showing where I'm from and where I am. So I graduated with a PhD degree from University of Michigan about 13 years ago. Doesn't seem to be very long. And right after that, I did a postdoc, a one-year postdoctoral fellow at Fred Hutchinson Cancer Research Center. So as you can imagine, I was trained in a very traditional fashion where I did methodology research in survival analysis, like human history analysis. And as part of my postdoc fellow positions, I start to move to like biomarker evaluation, risk prediction model, but all of those are within the survival area. So up until then, like all my research interests exclusively focused on methodology development. However, I always have a passion to help others, and like I'm particularly interested in doing consultation. So while I was still pursuing a master's degree, I actually did some research assistantship at University of Missouri, providing some consulting experience. So because of that, while I'm looking for faculty positions, I'm looking for a place where I'm able to provide a bit more collaborative contribution. So that's why Vanderbilt University attracted me. So recruited by Vanderbilt University in 2011 as an assistant professor, I stepped into an environment that is very different from where I graduated from. So Vanderbilt University has a very collaborative environment where Department of Biostatistics was housed within the medical center, and where our department is the only department that provides biostat services. So as you can imagine, there's a lot of collaborative opportunities. I think about 2 to 3 months after I was recruited, I was assigned to work with emergency department physicians, the whole department, more like we have a collaboration plan. And as you can imagine, there's a lot of use of EHR data by emergency medicine physicians where they can develop risk prediction models, or they look at charts and to do some research. And Vanderbilt is actually pretty famous for the EHR data because we have a very strong DBMI department. And so there's a very good scientific environment to get access to EHR data without any difficulty. So because of that, I get a lot of projects that actually use EHR data. That's my first exposure, actually, of using EHR data, where I know, there's a lot of data quality issues and we need to check it. But I still at a very on surface, it just use the EHR data to answer some clinical research questions. And after about 9 years later, I was promoted, and I will use the second slide to talk about what happened to me during this 9 years. So about 10 years later, after I joined Vanderbilt, I got a R1 grant as a multiple PI with the aiming to disseminate and implement a clinical decision support tool in our EPIC system using EHR data in real time. And there are statistical challenges. And the reason one of them I'm a PI is because there is a missing data issues that is different from the traditional missing data challenges. And I will talk about that a little bit in a little bit more details. So this, of course, getting R1 is not easy. It probably take me about 3 years working with the same group with multiple failures until we finally got it funded. But I think this is truly a change point for me, where getting exposed to a real projects where statistical methods can really contribute to addressing some real-time, like real-world challenges, it's really eye-opening. And we gain a lot of, like, I personally get a lot of experience. And all the other PIs, actually, they also said they gain a lot of unexpected experience through working this. And right now, this grant is still ongoing. And then 2 years later, I was appointed as the director of our CTSA Biostat Masters program, where I take more like a leadership position in terms of provide clinical support for clinical translational science. And at the same time, I also provide some leadership roles within the data coordinating center. So that's about my career timeline. So next I'm going to focus a bit more about my exposure with EHR data. So what happened to me between 2011 and 2020 was 30% of my time. And of course, those working in the medical center, our efforts are covered by multiple grants, right? So it's not like you have 100% of the time working on a project. You just have to split your effort. And for me, luckily, I have a not very small percent effort working within emergency department on various projects. So the one that I would talk about earlier is, once I joined Vanderbilt, I was actually introduced to participate in this R1 study with the goal of develop a risk prediction model. We name it Stratify. The goal of this risk prediction model is to identify acute heart failure patients who have low risk and thus can be safely discharged home. So right now, actually, there's about 80% of acute heart failure patients who was directly admitted to hospital. So as you can imagine, there's a lot of burdens. So by safely discharge even 5% of some low-risk patients will save a lot of money. So that is the motivation. But as many of you know, there's a lot of risk prediction model being developed. But how many of those are actually being used in clinical practice? It's not that many. I did some research in implementation disseminations. Actually, it takes about 17 years from the availability of clinical evidence to its clinical utilities. It takes 17 years. And out of those, all the risk prediction model, I think out of all the clinical real world evidence, probably half of those are actually being used. When it comes to risk prediction model, that percentage is even smaller. And why? Because there's a lot of challenges in implementing that. Using this particular model as an examples, so this model was built with an ordinal outcome, or 30-day adverse event ordinal outcomes, where we have 13 risk factors, which are routinely collected within 3 hours of ED presentation, like BMI, like some lab tests, and some medication history. So those who are familiar with EHR data, once you hear about the use of BMI, you probably will laugh because there's a lot of problem with BMI. Like there's a missingness in BMI, and weight and different union and height did not measure different at the same time. So we actually checked some existing EHR data showing there's about 30% of the eligible patients that has at least one missing risk factor within the Stratify formula. So which means even if we can, implement this model in our EHR system, there's more than 30% of patients can actually have a score calculated because of this missingness. So this missingness is different from the missingness we're talking about when you extract EHR and do some research. So this is more like a real-time missingness. So therefore, I propose some methods to address the specific issues, which leads to our dissemination implementation of Stratify R1 grant. So, and at the same time, I use the term implementations, which actually, this is the term that I start to get familiar with about 3 years ago after working with, people within this, the PIs of this grant. So I was introduced to the concept of implementation science not too long ago. And actually, I noticed like a lot of clinical investigators are quite interested in implementation science because that is really when you can put the clinical evidence into clinical practice. It requires a lot of implementation strategy development. And because of what we have done, between these two grants, my other ED physician collaborators, they leverage on what we have developed and where they also, get a PCORI grant about implementation of some other particular intervention within the ED area. So I was firstly involved in a PCORI grant, where it's multi-center clinical trials. And then using the implementation strategy, ideas we developed over developing the R1 grant, we eventually got this PCORI grant to implement this intervention. So the reason I put those two things together is, to me, is like using EHR data, and of course, when we talk about implementations, we also talk about extracting the EHR data, pre-implementation, post-implementation for comparisons, where there will also be different issues. So the reason I brought this up is, throughout, what happened to me between 2011 and 2020, over 9 years of period of times, there's a lot of things that I learned going through, this whole process with only 30% of my effort. I'm happy to talk a little more about details about challenges, but there's a lot of things that is beyond what a biostatistician usually do. For example, talking to different teams, and within this grant, we actually have probably 5 to 6 different teams across the entire campus, starting from the user-centered design for those who develop interface, to the knowledge engineering who actually talks about the artifacts, and to the health IT who does not do any research. They purely develop, product, but they don't do research. And also with implementation scientists, with biomedical informatics about building something within Epic. So there's different parties, and that is where I know, it's just like this is not like a traditional, statistical methods grant where you just focus on developing methods. It's involved a team science. So that's really an eye-opening experience to me, where I see, as a biostatistician, there's way more than things that I'm able to handle. And I learned a lot through that experience. So that's what I want to share with you today.
Thank you so much, Dandan. That's really insightful. And I think will be a great lead into our Q&A session. So we're going to move now to the moderated Q&A. Please use this time to start adding your questions to the Q&A function on Zoom. I have a couple of questions that I've prepared in advance that I'm going to ask first, but if you could go ahead and start thinking about your questions and putting them in the chat, then we'll have the opportunity to ask your questions as well. So my first question, and this really is like launches off of what Dandan was just now talking about, but I was wondering if you could share more about how your research collaborations with EHR data are different from studies using other data sources, particularly the makeup of your study teams, your research activities, and the role of the statistician. How do those differ from maybe non-EHR data research folks may be used to, and what kind of like communication and other collaborative strategies have you developed to effectively work in those teams and research projects?
I'll take that first, but I am gonna like completely build on what Dandan was just saying, because it really really resonated with me. And in my experience, my collaborations with the EHR data are not that different from my collaborations on other observational studies, but they are more highly multidisciplinary, and there's sort of a greater diversity of roles that people are filling on the team. So, often we'll have epidemiologists, MDs from various different specialties, informatics, programmers, occasionally somebody from healthcare, IT, sort of part of the administration. And then we also have this complicated, I'm constantly fascinated by the degree of hierarchy within the medical school. So we'll have this complex hierarchy of, the attendings and the fellows and the residents and students, and everyone has a really different skill set to contribute. And I would say the main thing that I think is different for my role as a statistician is just that the data and the questions that they want to answer with the data are complex, and often they're complex in ways that are subtle and not immediately obvious. So I think all the time about the fact that you can construct a data set out of EHR data that if someone just dumped it in your lab, it looks like a longitudinal observational data set, and you could analyze it using standard methods and probably come to a lot of incorrect conclusions. And so in order to kind of drill down into what's really going on in these data, you really have to listen. And I try to be very conscious of like checking my ego at the door, even if I'm working with people who have much less research experience than I do. They have much more knowledge than I do of the clinical context, the way the data are generated, how they interact with patients. And all of those things contribute to what ends up in the data set. So unlike an observational study that was designed where I could like read the protocol, in an EHR-based study, I have to sit there quietly and think carefully, like, okay, I'm trying to deduce what the like implicit protocol for these data looks like from what I'm hearing from my collaborators. So I think for me, it's sort of the soft skills of communication, listening, asking good questions are particularly important, because without them, you can make all kinds of incorrect assumptions and end up with wrong answers. But that said, I think like the role of the statistician is like at least equally important as it is in any other study. So I also try to think about making sure that I model the way that I want to be treated. You know, I don't want to be treated like a power calculator. I don't want the role of the statistician to be marginalized. So it's also important to make sure that I'm heard in terms of like having the final word on issues of study design and analysis. But I really try to approach it like a two-way kind of bi-directional learning opportunity. I'm going to learn from the clinical collaborators about the content area and the data, and they're going to learn from me how we can analyze it.
So I think, as I mentioned earlier, my experience is really, to me, it's pretty unique because I have to work with a diverse team. So starting with the proposal development, where it's pretty clear, like because I was involved in the original prediction model development. So when our original PI was thinking about, we want to, you know, work together on a grant proposal to implement it, and just naturally, like we work together. And then we start thinking about who do we need a third PI and who do we talk to? So we actually start by looking for people who can help us. So we first actually approached somebody from biomedical informatics department who's quite familiar with clinical decision support tool. And then later on, we talk about, oh, this may be for within the implementation, you know, area where there's some, you know, programs announcement, particularly talking about DNI. So then we approach to another internal medicine who is actually has some leadership within our medical center with for the implementation science area. So ultimately, we three work together and we submit 2 proposals, and the second time we got it. And still, the story does not end there, because even during the budget preparations, we talk about build, you know, clinical decision support tool, which I have no idea how to do it. So we need to talk to different peoples. It looks pretty intuitive. You find right people, you know, they can form a team to help you. But that's not the case until we got it funded and started to form the team. And I realized, you know, our original investigator who said, you know, she can help build the tool can only help, she only had the expertise, which is pretty natural, to do part of that, which is the knowledge engineering pieces, the work, identifying the artifacts with, like, use the FAIR standards for our prediction model. So the group who can actually build a tool belongs to our health IT, which is completely different department, and who was not in our budget at all. And we have no idea we need to include them in our budget. And so we just have to work around, and we meet with 2 project manager and 1 program manager to eventually nail down who should we work with. So that take about like a month or so. And then we also talk about, well, we need to develop a interface in who will do this for us. And well, there's some existing template we can directly use, but because we talk about the iterative user-centered design, which actually Vanderbilt has a center called Chris, who has the expertise of building interfaces, like infographics and the colors and what it looks like. They have the expertise, so we also approached them. So that center was also involved. And next step is after we talk about knowledge engineering, build health IT. And what I forgot to mention, this is multi-center grants, where other than Vanderbilt, we also work with Henry Ford Health System and Oregon Health, where both of them use Epic, which is nice, but their clinical workflow are a little bit different. So actually, during the proposal development, I read a lot of literatures, which is not statistical literature. I read a lot of papers about implementation science and how to do that. And I bring up the ideas we need to do a site visit to understand clinical workflow, because we need to understand when data was entered, at what time points, when they are available to be used, because we're talking about real-time use of the EHR data. One quick example is in our risk prediction model, we actually need EKG information, which, the order of that and the results read by the cardiologist, and there are different timestamps. So understanding and different health system have different workflow. And so it's quite different. So understanding different workflow, develop implementation strategy tailored to each site are actually critical. So I also, my another role for this grant is also to coordinate with different sites regarding the scheduling and understanding their Epic build. So it's also like eye-opening experience with me, because a lot of times I even don't understand the term they talk about. So I need to talk with their IT or informatics folks about things within Epic, like a term like about using the impression, the flow sheet and something like that. So just through this whole process, that's why I'm talking about like we have like probably 6 to 7 teams at a different time period. So that's really the skill sets that are required are very, very different. It's not something that you can get, during your PhD training. It's just you have to get involved and then be more proactive. I think I just step out of my comfort zone because of getting involved in this grant, because my role here and I just volunteer myself to get involved a lot of coordinations where I really gain a lot of experience, understanding the importance of not looking at data, but also understanding the entire workflow, the data flow, actually.
Thank you so much. I'm going to move on to the next question, and I'm going to start kind of mixing in some of the questions I had planned with what we have in the Q&A. We have some great questions. As you think of questions, please put them in the Q&A. So I want to go with this question we got from a first-year PhD student who's considering doing a dissertation using EHR data. And they say, as I delve into exploring databases that align with my research interest, I've received advice cautioning me about the complexity of EHR data. I was told that it often requires extensive cleaning processes and might not be the most time-efficient choice for a dissertation within the constraints of a PhD timeline. I would greatly appreciate any guidance or advice on how to effectively approach and handle EHR data for a PhD dissertation. And I might just add in here, it may be helpful to be real clear and maybe demystify a bit of how you get EHR data. Both of you are in environments where Dandan, I think you said and I received all this EHR data. But in other environments, the statistician's role might include more of that data compiling and cleaning. And so I'm sure your advice might depend on what type of environment the student is in. Yeah, as someone who works with EHR data, sometimes I forget that other folks don't realize all the things programmers and other folks do to get me that data. Yeah, which one of you would like to start responding to this question?
I can go first.
Yeah, go ahead, Dandan, you go.
Yeah, so I think this really depends on how you get the data, right? Like for me, for this particular grant, I have the budget to cover a core to extract the data for me. So it sounds rather easy, although there's also a lot of challenges. But in general, our university also provides some services that you can work with another core to extract the data. But for the different core, you need to work with a project manager to explain what you need. And as a biostatistician, if you don't work with a clinician or you don't work with collaborator, it's a little bit hard to tell exactly what are the ICD code you should use. So there's a lot of details that you don't want to dive into independently. You definitely want to start with having a collaborator. So I don't know, the people who ask, the student who asked this question, which level he is or she is at. But to me, if we are talking about cleaning EHR data, I think if the data were extracted properly with the correct inclusion-exclusion criteria already, cleaning the data requires setting up different criteria of data qualities, which maybe Rebecca can speak a bit more. But to me, I think it really depends on the size, the scale of your project. If you're talking about, oh, I'm just going to check some hypotheses, you can focus on data cleaning for just a subset of variables to start with. But ultimately, I highly suggest you collaborate with a clinician or whoever, a collaborator who has the clinical knowledge, because they're able to provide you what is the valid range for lab test, what are the plausible or implausible values as part of data cleaning. And actually, for our risk prediction model, we incorporate that into our clinical decision support tool. Like when we build this tool, we specify for each continuous variable what are the plausible ranges. And if it's out of range, then we will flag it or we'll set it to missing, like certain rules that you need to determine as part of data cleaning. So to me, it's really very worth the time to do that. If you don't have a collaborator, you probably do not. You definitely want to have to work with somebody together. And also depends on if you want to use that for 1 or 2 projects, or you have like a serious project that you have some idea to work on. It really depends on the complexity of your projects. I think Rebecca may have some additional insights to this.
Yeah, I agree. And I was thinking, as you were speaking, I was thinking there's so much of my answer and so much of what you were saying involves like, it depends on like what resources are available at your institution for accessing EHR data. Like here at Penn, we have data navigators who will help you to figure out where the data that you want actually live within the EHR and do some of the extraction for you. Other places have a data warehouse that you can go to directly and access research data. But I think often when we say EHR data, people who don't have experience working with it don't realize that like that can mean many things, all the way from sort of the raw healthcare operations data through to the very harmonized, processed, data cleaned data set that you actually do research with. And so like, depending where you're going to get involved on that continuum, the effort that you will devote to the data cleaning process is really, really different. And in fact, like you can't, as a researcher, like I can't access raw operations data. So there definitely has to be someone who's going to be an intermediary. That said, like I'll just reemphasize what Dandan said about having a collaborator. Like you really, really have to have a content area expert working with you, because the data are so huge and there are so many nonsensical things that you can do with them if you don't know, how they get into the database and what are the typical lab tests that are recorded, or like why would we sometimes see this and not that. Is really, really critical to have that expertise. And related to that, I would say, I think you can, sort of putting a more positive spin on it, you can very much reduce the complexity and messiness of the data by focusing on a specific scientific question and a specific use case. So when we think about EHR data broadly, it's like, oh, they're super complicated and it's a mess. But if you have a specific question that you want to answer, then you can say, okay, to answer that question, the 5 variables that I need are, these things. And then you can start to really operationalize things and get precise and figure out whether or not there's adequate information available to like populate those specific variables. So I think you can very much reduce the complexity of the problem by being specific about the context that you want to focus on. For me, like having a concrete use case is like the only way to make things manageable. Otherwise, it just spirals out of control.
Great, thank you both so much. One of the questions I had planned actually merges well with one of the questions in the Q&A. So the question I had planned was that just EHR data are messy and imperfect, as we've been discussing, and asking about how you bring both rigor and pragmatism to EHR data studies to generate valid and meaningful results. And one of the questions in the Q&A was someone saying they'd love to hear some more stories on those thorny decisions that have to be made when it comes to trade-off between practicality and validity. So do y'all have any general thoughts or specific examples that are illustrative to share?
I feel like the farther I progress in my career, the more inclined I am towards finding practical solutions, and the less inclined I am towards finding precise and exact solutions. I think part of that is because working with data that are messy, you realize that like with any given EHR data set, you're going to be dealing with like selection bias, missing data, measurement error, informative observation, probably a bunch of other problems in any specific context. So I'm almost always asking myself, like, what's the most important problem for this particular question? Like, what's the key threat to the validity of my research results, or what's the key source of bias? Because I don't have time to develop statistical methods that address every single problem simultaneously. Methods don't exist to deal with that. And it might be that some of the issues that I'm thinking about are trivial in a particular setting. So like recently I was working on a problem where I was really worried about missing data in inclusion-exclusion criteria and how we should handle that. If you do multiple imputation, for instance, for inclusion criteria rather than covariates that are in your already identified study sample, that sort of results in more complicated problems than a more traditional MI setting. So I was working with my postdoc, and I was like, what do we do about this? You know, should we use this method or that method? And then we realized that like, actually the number of people who would be potentially included if we imputed their covariates versus excluded if we excluded everybody with missing data was really, really small. Like made no difference. So I was like, okay, this is a theoretical problem, but it's not a practical problem. So that kind of thing, and sometimes it takes like doing a small simulation study or some kind of quick applied methods investigation to figure out what's the most important problem or the most important threat to validity in a given context. I like doing those. I think it makes the research more rigorous because I've given kind of formal thought to what the big problems are. And also you can write some nice, I think very useful to other people, practical methods papers that just sort of compare, you know, if we use A versus B, or if we make choice A versus B, and or those become nice sensitivity analyses for the paper itself. But I feel really strongly that it's valuable to get an approximate answer to the right question. There's absolutely no value in getting a perfect answer to the wrong question. So if we have to use a method that the data satisfy all of the assumptions of, but that answers a question that's not the scientific question of interest, that to me, it's a total waste of everyone's time. So I'm all in favor of doing the best we can with the data that we have in hand and the practical solutions that we have in hand, really guided by the scientific questions of interest.
Think I can share one or two examples to talk about my opinions. I completely agree with what Rebecca just mentioned. There's a trade-off in terms of how rigorous you want to handle all the data quality issues versus what is more like feasible. So in our study, as you know, we extract EHR data. Like we need to first validate our risk prediction model. So validating the model require extracting both the outcome as well as all risk factors. So after we got this data extracted, the first thing we did is to understand our EHR population and compare that with our original cohort that was used to develop the tool. And through this process, we identify a lot of data issues, like for example, BMI. By doing using various visualization, we know these older ranges is ridiculous. Like BMI can be as low as 1 or as high as 100. And obviously there's a unit issue, like the unit was not used consistently. but there's like thousands of records. I'm not able to ask my PI to check each of the BMI and the crack. It's just not feasible. And a lot of BMI are calculated using like a last value carry forward for heights, because we know for adults, height does not change that much. And for weights, you just have to make some assumptions. So even after we use that approach, there are still some very implausible values. And it's just not realistic and not good use of our time to do all the chart review to crack those for like a couple of thousand records. But so for this type of data quality issues, what we did is really just, understand the proportion of implausible values and try to say whether we can truncate within the plausible ranges and run a couple of sensitivity analysis to see how it influences our results. But on the other hand, our adverse event rates, our outcomes, the adverse event rates is about, I think, 33% within the EHR data, where our original cohort is about 12%. So there's a huge difference. So we started wondering about, is that we extract the data in the wrong way and why? And then by looking some of the cases, we understand, oh, probably the original cohort was done by having patients providing consent. So I think you can imagine with the ED, those who can provide consent, they are less sicker, right? They're less severe. So there is a natural selection bias when you develop a model, by enrolling those. So those are reasonable. And then we will also look at the same thing for Oregon Health, OHSU. So where we notice one of the components that define this primary outcomes is its prevalence, where its event rates is actually unstemming. So the event rate is zero, like there's no unstemming, which is not possible. So those are the things that you cannot miss. Like you will have to be very cautious and rigorous, especially when it comes to defining the outcomes. So after a couple of months exploration, what we found out is unstemming was defined by using a lab test called troponin. However, troponin has a different assay called high-sensitive troponin, which Oregon Health use on a regular basis. They don't use troponin, they use high-sensitive troponin, where Vanderbilt use troponin. So we use a cutoff for troponin to define unstemming. Oregon Health doesn't have, they don't have troponin, so they use high-sensitive troponin, but we did not provide a cutoff for high-sensitive troponin. And the people who extract the data didn't let us know. So just after a couple of months communication, we figure out, oh, this is what happened. So we just, work with their site PI and to talk about what is should be used as a site-specific cutoff for high-sensitive troponin. But it takes a couple of months, but I think it worth the time because it matters, because that's our outcomes. You have to have your outcomes clearly, correctly defined. So to me, those are just two different examples showing what you should focus on, what are things that you can probably balance.
Great, thanks. We're running short on time. I want to get one more quick question in here. I really appreciate everyone's questions in the Q&A, and sorry we're not going to be able to get to all of them. But just really quickly, are there one or two areas that you all see as growing areas of opportunity for statistical leadership in EHR, something you haven't gotten to talk about yet today?
One thing I'll briefly mention that we haven't talked about at all, but I think is like a huge growing area, is the role of EHR data in regulatory submissions. So a number of years ago now, the 21st Century Cures Act was passed in Congress, and it basically said the FDA has to figure out how to incorporate real-world data into the evaluation of new medical products. So FDA has been working on that and refining their thinking. And you can imagine that at the same time, the pharmaceutical industry is like, how can we capitalize on this and start to incorporate these data into our regulatory submissions? So there's so much interest right now on the pharma side and also on the FDA side. It's definitely a thing that's going to become more and more a part of the drug approval pipeline over time, and it's fraught with like all kinds of statistical methodological challenges. So I think as people are thinking about careers, if this is something, if you like the messiness of the data, you don't want to just do like clinical trials and like clean data, I think there are a lot of opportunities in government and also in industry for people who want to get involved in using EHR data as part of the regulatory approval process.
So one area I can think about, again, is because, I was work at Vanderbilt, and Vanderbilt have some resources called learning health system. I think this is not a very new term, but it's pretty popular terms where you learn from your data and you apply what you learned from the data to a clinical practice. So this learning health system, and also people use that to conduct programmatic clinical trials, is really just leverage on the EHR data in a pragmatic way, like extract EHR data, analyze it, and then and in a way that you don't directly do like a clinical trial, but it's more like a pragmatic clinical trial. So we have a lot of research, like we conduct a lot of learning health system type of research at Vanderbilt. And I do see those are a lot of potential opportunities for biostatisticians, because all the learning health system type of research projects require biostatisticians' involvement to have a study design and to talk about what is a valid definition for the outcomes that can be derived from EHR data, what are the data availabilities, and there's a lot of things that I think can be covered. So that's one. And the other one is, of course, because I work in this, clinical decision support development, I see the expanded research proposals. Because I think my ED physician and their collaborators sees the successfulness of our grant proposal, there's more and more implementation science, focused proposals where they talk about building, some clinical decision support tool, use the EHR data, leverage on the IPIC system. So I think that's another potential area that I think we can think about.
Great, thank you both so much. Those are three really interesting ideas for us to leave folks with. Thank you, Dandan and Rebecca, so much for joining us this morning and for sharing your expertise. Thanks to the audience for listening. If you found this conversation helpful, please share the recording with your colleagues. It will be made available to you. Special thanks to those behind the scenes that make this webinar happen, including Megan Glenn, Mary Ryan, Natalie Dean, Amita Manatunga, and Jim Rosenberger. And we hope that you'll all stay tuned for more events like this from the COPSS NISS Leadership Webinar Series. Thanks again, everyone, for joining us.
Thanks, Yates. Thanks, everyone.
Thank you, everyone.